So I'm happy to present Dorothy Menefee. She is originally from Kansas, but in recent years from Texas. She did her PhD in Texas A&M University, and she is currently a postdoc with the USDA ARS. Thank you for agreeing to talk with us at this time in Texas. We appreciate that. And please, Dorothy, go ahead.
Okay. Hi. As said before, I'm Dorothy Menefee. Right now I'm a postdoc with the USDA ARS, and my talk is evaluating and simulating evapotranspiration in Texas agroecosystems. So about me, I'm originally from Spring Hill, Kansas. I did my undergrad at Kansas State University, and I also did my master's degree there, which was on the thesis was the anthropogenic influences on soil microbial properties. I did my PhD at Texas A&M with a dissertation on modeling evapotranspiration and carbon flux in corn and cotton. I currently work for the USDA ARS in Temple, Texas. So a bit about us at the USDA ARS. We are the research wing of the USDA. It's sort of equivalent to your organization in Israel. We have 90 locations across the United States, and our location in Temple, Texas is the Grassland Soil and Water Research Laboratory. Our location is part of the LTAR network, or the Long-Term Agricultural Research Network, which is a sort of subcategory of 18 sites, which focuses on long-term management effects on various agronomic properties like productivity, profitability, and ecosystem effects. So our site, we primarily grow corn, cotton, wheat, teff, safflower, and then we have a lot of hay pasture and some cover crops like Austrian pea and mustard. Our current focuses are things like crop phosphorus use, tillage practices, greenhouse gas emissions, which is my main focus, precision agriculture. We also do some work with remote sensing and agricultural disease control. So kind of after that introduction, I'm going to sort of move into my main talk, which is on simulating evapotranspiration using DSAT. So DSAT is the Decision Support System for Agrotechnology Transfer. This is a modeling platform that was developed in the 1980s, originally for legumes like soybeans and peanuts. And right now it incorporates 42 different crops. This model has sort of a modular style approach where you combine multiple different types of models and different systems to sort of create a output. So this diagram here gives a good sort of flow, a simplified flow of how DSAT works through the three main submodels: the plant submodel, the weather submodel, and the soil water submodel, and they all combine together for one output. And that's definitely how the evapotranspiration simulation works within DSAT. So in addition to DSAT, I also have eddy covariance measurements. So eddy covariance is a micrometeorological method for analyzing gas flux. It's commonly used with water vapor, CO2, and methane. And the flux will be the covariance between the gas concentration variation and the variation of the vertical wind speed. And this technique requires a really large uniform area. And then the sum of our water vapor fluxes will be evapotranspiration. So the main objectives of this study are to simulate evapotranspiration in dryland corn using the DSAT system. We also have a similar study where we simulated cotton evapotranspiration, but I'll just be presenting corn to you today. Our goal was to compare the simulated evapotranspiration from eddy covariance to that was simulated using DSAT. And our goals are to sort of further understand the weaknesses in dry land evapotranspiration modeling, because that's one of the areas where DSAT tends to struggle, is with simulating ET and other sort of water parameters in dry land systems as opposed to irrigated systems. So our study site for this is the Texas A&M Agricultural Research Farm. It's located near College Station, Texas. There's sort of, if you see on the map of Texas, there's a red highlight of the county where it's located. And then we have just a diagram of the field. It's this large area that I outlined in yellow. The star in the center is the location of the eddy covariance tower. And right along the field is the Brazos River. So that kind of adds a bit of an edge to the field. So the climate is humid subtropical. We get about 1,000 millimeters of rain a year with an average temperature of about 20 degrees Celsius. The typical growing season for corn in this area of Texas is typically March through July. And the soils at the location are vertisols. And this site is just conventionally managed with traditional tillage and fertilizer practices. And the eddy covariance tower was established in 2017. Here I've given a list of just the varieties and planting dates. So the planting dates were all really about the same every year, just a few days off. In 2019, we ended up harvesting a bit late, and that's just due to delays of getting the machinery in. The crop was mature at about the same time as it was in previous years. The only other difference with 2019 is that they weren't able to perform the full tillage practice that they normally would due to unusually wet soils in the spring. So our instrumentation for the eddy covariance system consists of a sonic anemometer and an infrared gas analyzer. So our sonic anemometer gives us wind speed and the wind direction components, and it's the Campbell Scientific CSAT 3D. And then we have an infrared gas analyzer from Lycor, and that gives us both water vapor and carbon dioxide, but we're going to be focusing on water vapor. So both of these instruments are connected to a Lycor SmartFlux, which does a lot of the data processing in situ using the Eddy Pro 6.2 software. And that will take our 10 Hertz initial data and convert it to 30-minute summaries. So when we go to collect the data, it's already been put into 30-minute summaries instead of the original 10 Hertz raw data. In addition to the eddy covariance instruments, we have a number of meteorological instruments at the site. So we have temperature and relative humidity, precipitation, net radiation, irradiance, plant available radiation, soil temperature, soil moisture, and soil heat flux. All of these other instruments give us additional data that helps us both with modeling and with checking the eddy covariance data. So our eddy covariance data is initially processed through the SmartFlux, which is shown here, this device here, which is directly connected to the instruments, and it has the Eddy Pro 6.2 software installed. And it performs a number of standard corrections, like coordinates rotation and things like that, to just make sure that the data is good. It does an initial inspection, and then after Eddy Pro has gone through the data, I do a manual inspection myself, and any erroneous data I remove, and I gap fill using the Max Planck Institute for Biogeochemistry's method. And this is an R-based program, and you can kind of see an output of that here. And I'm using the nighttime method, and version 73 using R 3.4.1 of the gap filling software. And after gap filling, I calculate an energy balance and then perform an energy balance correction. So I plot LE plus H versus RN minus G to get the slope. And that can give you an idea of how well your eddy covariance system is capturing all of the energy flow within a system. And there's always a gap. RN minus G will always be a bit more than LE plus H in almost all systems. It's pretty rare to get complete energy balance closure in an eddy covariance system. So typically energy that was missed or captured by RN minus G and not by the eddy covariance system can be reallocated to LE and H based on the Bowen ratio, which we calculate manually. So once we have the eddy covariance data calculated and ready, we can work on setting up our DSAT simulation. So in the experiment file, I have it set to series maize, which is one of two methods for simulating corn growth in DSAT. The other is EXIM, but series has a bit of a longer history with DSAT, and there's a bit more literature behind it. So we set the simulation to start on March 1st in every year and set it to end on crop maturity. So it's important that we start the simulation a few days before the crop is actually planted, because this gives the soil water module a bit of time to sort of calibrate itself. We have a weather file where we set up with a bunch of daily information: minimum temperature, maximum temperature, precipitation, relative humidity, vapor pressure deficit, wind speed, and irradiance. And then we have our soil file where we enter our clay content, our nitrogen content, sand content, organic matter, and depth to water table. And then finally we have our file T, where we enter some of our collected plant growth data from throughout the season. And in there I entered biomass, leaf, plant height, and leaf area. So how does ET simulation in DSAT work? DSAT simulates ET in two steps. The first step is to simulate potential ET, and to do that, DSAT uses one of two methods, the Priestley-Taylor/Ritchie method and the FAO 56 method. Both of these methods use weather data alone. They do not incorporate any information from the crop module, and this is just a potential ET using a grass reference. So in the Priestley-Taylor-Ritchie method, it's a bit simpler. It only uses, I believe, temperature and humidity, and then the FAO 56 method adds in wind speed and solar irradiation. And both of these methods are sort of the jumping off point. And to sort of compare them, we actually used both in this study. So I'm looking at both methods and comparing and contrasting them to each other. Our hypothesis is that the FAO 56 method might provide a bit better simulation, just because it includes the wind speed and solar irradiation into its estimate of potential ET. Which can usually give you a bit of a more precise estimate. So once DSAT has calculated potential ET, it then partitions that potential ET into potential evaporation and potential transpiration using the leaf area index. So this is where it begins to pull in data from the plant model. So it's going to pull in the leaf area index of the exact day that it's calculating, which is sort of based on what I input from my measured LAI, but since I don't have measured LAI for every single day, DSAT simulates LAI for every single day, and then we'll use the LAI of that day. And then finally, once the potential evaporation and potential transpiration are calculated, DSAT will then generate crop ET using the root growth and soil water data. So this is pulling, again, both from the soil water module and from the plant growth module to get the root information and the soil water information. And it has a maximum crop coefficient, or KC, of 1.0, which we'll discuss a bit later, because that creates a few potential problems when we are getting our results from the model. So on to some of our results. So this is just the observed evapotranspiration for the 2017 growing season. The black dots are daily measured ET values from our eddy covariance system, and the blue fills are just precipitation for that day. So for 2017, we have fairly constant ET throughout the growing season. We do have a bit of a spike towards the end after a couple of rainfall events where it's mostly soil evaporation. But throughout the main growing season, things stay pretty steady, even after this really large rain event. You do see a bit of a spike here after that, but it quickly goes back down. And this is for 2018. In 2018, we had fairly low ET at the beginning of the growing season. And you see a strong response due to this really large rain event here. And then ET quickly rises for the main portion of the growing season. And we see a similar trend to in 2019 as to what we saw in 2018, sorry. Again, with really low ET at the beginning of the growing season. And it's kind of important to note that 2019 was a bit of a wetter year. If you just look at the sheer number of rain events, you can kind of already tell that just by looking at the graph. But 2019 was a bit of a wetter year, especially in the early growing season. So looking at observed ET for all years, I've combined just the observed ET for all 3 years onto one graph here. With 2017 in green, 2018 in blue triangles, and then 2019 in gray squares. So the biggest difference that I notice is that we have the really high early growing season ET in 2017, and you don't see it as much in 2018 or 2019. So our cumulative ET in 2017 was less than our cumulative precipitation, and we see the same thing in 2019. However, in 2017, our cumulative ET was greater than our cumulative precipitation, indicating that soil moisture is fueling some of the crop growth, which can happen when it's a bit of a drier year. So stored soil moisture is what is allowing the observed ET to be higher than the observed precipitation during that time period. So the next step for us was to simulate some of the phenological properties of the crop. This can give us an idea of how well DSAT's doing, and it also allows us to configure the cultivar parameters, which are some of the genetic crop parameters that DSAT uses. And to do this, we use our collected growth data. So you have here, I'm just showing leaf area index for all 3 years, with the blue squares representing our measured points and the black dots representing our simulated points after we have calibrated using the generalized likelihood uncertainty tool, which just uses our plant growth data to get an idea of what the correct genetic parameters should be. So finally, moving on to some of our evapotranspiration simulations, which are sort of the main goal here. We start by comparing the potential ET, so the first output of DSAT, to our reference ET. So this is an estimated potential ET that we calculate manually using just the weather data. And that's what you see in the first column here with the green dots. So potential ET from the Priestley-Taylor method, PT, on top, compared to below it, the potential ET of the FAO56 method, and both of them compared to the reference ET from the site. And then on the right with the red squares, we have the measured ET compared to the crop ET output from DSAT. So this is the ET output that has been corrected for the actual crop growth model. And what we see here in the beginning with the potential ET is that we have a really good correlation between calculated potential ET and our DSAT's potential ET. And then when we move on to looking at the actual crop ET, we see a lot more variation between the two, the measured and the simulated. We have a bit of a better correlation, or R-squared, with this Priestley-Taylor method than we do with the FAO56 method. However, we have a better sum with the FAO56 method. So throughout the cumulative measured ET for 2017 was 440 millimeters. And the FAO56 method estimated that it would be 474 millimeters, and the PT estimated 516 millimeters. So both methods overestimated actual ET, but the FAO56 method was a bit closer. Moving on to 2018, we see a really similar trend in 2018 compared to 2017. So again, we have a pretty good correlation between the DSAT potential ET and the reference ET that we calculated manually. And then once we move on to the crop ET from DSAT compared to the eddy covariance ET, there's a bit more variation. This time, the R-squared is a bit better than it was in 2017, but we still have the overestimation. And it's important to note that this was a bit of an unusually dry season where evapotranspiration exceeded precipitation during the growing season. And once again, the FAO56 method produced a cumulative estimate of ET that was much closer to what was observed. So in 2018, we had observed 362 millimeters of evapotranspiration during the growing season, and the FAO56 method predicted 432, and the Priestley-Taylor method predicted 452 millimeters. So finally, 2019. The 2019 results are a little bit different. So in 2019, we actually had better correlations with the FAO56 method, and we also had a bit of a closer match between the estimated sum with the FAO56 method in 2019. But once again, as with the previous 2 years, the DSAT system overestimated actual ET, this time by about 50 millimeters for the growing season. For both methods, there was really only a tiny difference in estimated sum between the two methods. So to kind of get at what's going on within DSAT, our next step was to look at the crop coefficient calculation. So this is, so you calculate the crop coefficient by dividing the estimated or actual crop ET over the reference ET or potential ET. So we did this for our observed data. This is all 3 years. We did this for all 3 years, but I'm only showing for 2017 because the results are really similar across all 3 years. So for the crop coefficient for our observed, you see we have a maximum crop coefficient of about 1.4 observed. However, with DSAT, the crop coefficient is hard-coded at a maximum of 1.0, so that's why you see this really flat line that looks somewhat artificial. You wouldn't really see that in a natural crop, and it's really common, especially for some of your high-yielding modern cultivars of crops like corn and sorghum, to have actual crop coefficients above 1.0, which is a potential problem with the DSAT system with both methods, that the crop coefficient is hard-coded at 1.0. So this essentially sets a maximum crop coefficient, and it alters the results of the model because the coefficient between the coefficient affects the way that the model converts potential ET into actual ET. And since the coefficient is a bit off, that can create some issues. So comparing our simulations, we used a few statistical indices. First is the mean squared error, which most people are familiar with. We also use the coefficient of residual mass, which gives an idea of over or underestimation. And of course, as we already saw, we have overestimation in every single year, which is indicated by negative CRM values. So CRM, a negative value indicates overestimation, while a positive value indicates underestimation. And then finally, we use an index of agreement. So this is a parameter that compares the modeled to the measured, and it's a statistical parameter that's really specialized for use in simulation. In an ideal, d-index would give you 1.0. However, the closer to 1 you get, the better. So by d-index and by our root mean squared error, by all of them, our best simulations were in 2019 with both methods. However, the 2019 simulations with FAO56 were the most accurate of all of our simulations, and the least accurate were those in 2018, where we had the largest overestimation and the greatest error. So in 2018, that was our really dry year. So it was a little bit different than the other 2 years. What is interesting is that 2019 was a bit of an unusually wet year, but it's also a year where our tillage practices had recently changed. So in 2019, we did not do full tillage because the farm implements were not able to get to the field. And this is just a bit interesting that the model did best on a year when we had just changed tillage practices, which isn't something that would necessarily be expected. So some of the potential causes for this, some of these model weaknesses, or why the model is heavily overestimating ET. So one of the major weaknesses within the modeled system is that it doesn't capture all the processes that are going on within a system, including soil variability and the impact of pests. So for instance, one of the major problems that we had in this field, in the actual crop field, was that we had a lot of feral hog damage. So where we are in South Texas, there's a large population of feral domestic pigs, and they destroy large sections of crops every year, including the field that we were working in. And large areas of the field that would have been captured by eddy covariance were essentially flattened. And you're going to have very different ET from the area that's been flattened by feral hogs versus the corn that's still growing. And we also had quite a few weeds, especially late in the growing season, which isn't something that is ideal, but it's something that happens. We probably don't have much of an influence of weeds throughout the main growing season because we didn't have very many in the early portion of the crop growth, but once the crop neared maturity is when we started to have a few weed issues. But most of our weed problems were after harvest and after maturity, so that would not be reflected in the model. One of the biggest issues that I noticed was that DSAT treats all clay minerals as one-to-one clays. So there are a number, clay minerals are a large category of different types of minerals, and they have different ways that they interact with water. So DSAT sort of assumes that that when you enter clay content into the model, it's going to treat that clay as if it's a one-to-one clay, while most of the clays that we have on our site are actually 2-to-1 clays. This is just referring to the mineral structure. And these different types of clays interact with water differently. So 1-to-1 clays don't expand or contract. 2-to-1 clays do. They can absorb a lot more water than a 1-to-1 clay. However, they hold on to that water a lot more tightly. And they actually expand when they absorb water as they're swelling up. And we're going to kind of talk a little bit about this in a bit. And then secondly, third, the ET simulation tends to be more successful in irrigated systems. And this is something that we noticed in the literature, and it's pretty well documented just across the different areas where people have tried to apply DSAT for simulating ET and for simulating other parameters, is that it works a lot better in an irrigated system where you are having constant and I guess very consistent water input into the system. So you're hardly ever going into the more complicated water interactions that happen in a bit drier soil. And it's also a bit of a more consistent system. However, this is a potential problem for areas where dry land production is the main method of crop production. So in the area of Texas where we're at, where this study took place, almost all of the corn and cotton grown is not irrigated because we usually get a decent amount of rainfall every year that the irrigation isn't necessary. So that creates a potential problem for attempting to model what's going on in this part of the country, simply because most of the models work best with irrigated systems, and most farms in this area are not irrigated. And then finally, as we discussed earlier, there's a problem with the KC calculation. that might be partly responsible for some of the differences between what's observed and what's simulated. So some of our ideas for how to improve this, one is to use, first is we're going to do the same exact stuff with cotton and can complete the KC analysis. That's not really an improvement, but that's something that we're still working on. Part of why I presented just corn to you is that we're still doing some data calculations with cotton. The big thing that I want to do to improve the model is to incorporate the high prop. So this is a instrument from Meter that calculates a soil moisture release curve over the course of about a week. And just a few weeks ago, I went and collected soil cores to use with this. So this device will allow us to have actual data points for the soil moisture curve. The model that I showed you, the soil moisture curve was simulated by DSAT using the soil texture as input. However, it is possible to go in and change that as a DSAT user. And that's what we are planning to do when we calculate our actual soil moisture release curve using the high prop. And I'm anticipating that what we actually calculate will be a bit different than what was simulated through DSAT, just because we have a 2-to-1 clay, which the vertisols that we have can act a little bit differently with a soil moisture curve. So that I think will be interesting and will hopefully improve some of the model when we incorporate that into the model. And we're probably going to start using this in January. Right now we're in the middle of a lab remodel and moving a bunch of instruments around. So our current goal is to set up the high prop in January and start running it then. So some of the other research that I'm doing, that was kind of an overview of our work with eddy covariance and DSAT modeling, and I'm going to kind of pull in some of my other work just so that you can get an idea of what we're doing at ARS and some of the other work that's going on here. So right now, one of the big things that I'm involved in is a flux climatology of all LTAR sites. So I have here a map of all LTAR sites across the country, and we're the TG site, so that's Texas Gulf. So what I'm trying to do is I'm trying to compile eddy covariance data from all of the sites. So as of right now, every site within the network has eddy covariance towers installed. So we can combine all of that data to get an idea of what are the trends in carbon and water vapor flux across a wide variety of sites, trying to get an idea of what are the influences of climate versus management practices. Because one of the nice things about the LTAR network is that all of these sites have similar management practices, but they're scattered across different climates. So we have essentially 4 different types of management practices that occur at all sites. One is sort of aspirational or ideal crop production, which means that we're using most of the more recent best management practices like variable rate fertilizer and minimal tillage and cover crops, and then conventional where we're just doing standard tillage and fertilization practices. And then we have aspirational grazing, where there are cattle or other livestock that are grazed using more recent best management practices, typically rotational grazing. And then we also have conventional grazing, where the animals are just grazed as they are typically on most farms. And finally, most sites have a remnant of native prairie or other native ecosystem that they also can collect data from. So our site at the Texas Gulf, we have 3 eddy covariance towers. We don't have any in the cattle grazing right now. We have one in aspirational crop production, one in conventional crop production, and one in the native prairie. So I'm looking to sort of compare the tower data from multiple sites across the country to sort of tease out what is from management and what is from climate. So additionally, I'm looking at the effects of tillage practices on greenhouse gas emissions and soil microbiology, and that's directly at this site and at a site in Indianapolis. So I have 2 sites that I'm working on with that. And then we're going to be incorporating greenhouse gas emissions into our moisture curve study with HYPROP. That's not going to be part of the DSAT portion of HYPROP, but we're going to be doing greenhouse gas emission monitoring throughout the soil moisture curve evaluation with the HYPROP. And then again, I'm still looking at carbon flux from corn and cotton production. So you saw the water vapor data, but I'm doing similar work with carbon flux, sorry. And then I've also done a bit of work simulating GPP using satellite remote imagery. So this is the field where we did our DSAT study in a reverse false color composite near infrared. And this was taken after corn harvest. So you can see the corn is mostly dark. That's because there's not really much plant growth. The bright red that you see in the field right below the cornfield is actually cotton, because at this point, when this image was taken, cotton was really still growing, but corn had been harvested. So then this is just a little look at some of our flux climatology work, which is the first thing that I talked to you about. So this is the standard climate curve with the average in the thick blue line in the center is the average. And then the thinner blue lines with the shaded area represent plus or minus 1 standard deviation. So this is the Kellogg Biological Site in Michigan. So looking at the standard climate there. And this is a summary of 18 site years, observed precipitation and temperature by month. And then we can also look at some of the carbon fluxes from that site. So this is continuous corn on the left, and then brome grass prairie on the right. So this is the conventional agricultural site, and this is the native prairie site. And you can see there is a difference. And since we know that these are both at the same location, we know that the difference is observed, likely due to differences in management and not climate. So obviously with the conventional corn, we have a net carbon emission throughout a larger portion of the year. And then we have a very sharp carbon uptake just during the corn growing season versus the native prairie. The carbon uptake happens over a longer period of time, which is kind of interesting. So, and we also have a bit more variability later in the year with the native brome grass prairie. And then this is from our greenhouse gas emission work. So this is from the Indianapolis site, which I have a bit more data on right now. This is actually not an LTAR site. This is a different location. What you see here are 4 different treatments of continuous corn. Treatment 32, where we have the high methane emission, is a conventional till urea fertilizer system. And then here, treatment 23, where we have net CH4 uptake over 36 site years per treatment. So it's a pretty good amount of data that we have compiled here. We have net methane sequestration, and this is a no-till system with urea fertilizer. And then these 2 treatments where we had essentially 0 methane emission or uptake, it was a little bit of sequestration here in treatment 31, but it's not statistically different from 0. These are both conventional till treatments that use a manure fertilizer, and I'm working on similar data to this from Texas. So finally, I'd like to sort of conclude this with a few acknowledgements. I'd really like to thank some of my colleagues here at ARS, especially Hal Collins and Doug Smith. Hal Collins, Doug Smith, and Russ Scott, and then Nithya Rajan at Texas A&M. And then all of our farm managers and assistants whom this would not be possible without. So just managing these farms, there's a really helpful crew of people that assist with planting and harvesting and getting all the instruments in and out. And then our LTAR network. And then you can see here just I have a few images of how we're doing the greenhouse gas sampling, the static chambers. Thank you. Are there any questions at this point?
Thank you, Dorothy. If anyone has any questions, please unmute yourself and ask a question. I'll go first until you master up the courage. Dorothy, you've shown differences between the reference evapotranspiration that you're calculating yourself and the FAO 56 calculation in DSAT. Why is there a difference between the two? Are they not fed from the same data?
They're fed from the same data. Most of I think it's using a slightly different equation, and the DSAT system it's incorporating some of its own modules and its own reference points. So sorry, so the DSAT system uses the grass reference in its FAO 56 calculation. And I believe that we felt that the alfalfa was more applicable to the site that we were at. So we used a slightly different one just because we felt that that would be a bit more accurate compared to what DSAT was doing. But it was really very close between what we calculated and what DSAT calculated using the same data.
So had you used the grass reference, it would
yeah, we used, and we
still
and we still got a little bit of difference just because DSATs we're both working on a daily time step, but there's just a few variations in the way that things are calculated, and it's not using the exact FAO 56 equation. DSAT has a few modifications that they put on it to make it fit within their system.
Okay, thanks. More questions?
I have a question. Hi, Danny Kurtzman, Volcanic Center. As for the cracks and the vertisols, do I understand you assume you get less evapotranspiration because you lose some water that flows in preferential flow down below the root zone through the cracks?
That's part of it. And also the potential for the soil is a bit different. So water is a bit more strongly attracted to the mineral. So it takes more energy to remove water from the clay mineral than it does a different clay mineral, particularly the water that is inside of the clay mineral. So the 2-to-1 clays, when they expand, water actually gets inside the mineral structure, and that water takes a lot more energy to remove than water that's attached to a typical clay.
Okay, thank you.
Marcel, did you have a question? I see you unmuted yourself.
Well, I'm not sure I know exactly what to ask, but I'm worried about the energy balance closure. You've shown that you are systematically underestimating the closure in the covariance method. I'm worried about the high variability that you are observing. How do you explain that? I'm especially worried about the large number of data where you have the eddy correlation sum being below 0. Do you have a comment about that?
Yeah, that's so it's not ideal, but it's pretty typical of eddy covariance systems just across the board. Part of it is that the eddy covariance system is a bit fickle sometimes, and it can, especially if you're having a recent rainfall event, or if there's just anything that's a bit abnormal, it might give you anomalous readings, which is why you do the energy balance closure. And it's only capturing water vapor flux. It's only really measuring the water vapor flux. The way that it estimates the heat flux is a bit different, so it's not going to quite capture that as well as it should. So it's just a best practice to do the closure. I'm sorry, I'm not completely sure on all of the reasons why it has that, but it's something that almost every eddy covariance site does.
So it's
It's one-
typical that you keep the data within those half-hour periods when it's raining, because you're using the open path core, right?
Yeah, we try to remove most of them, but even when you remove most of them, you're going to get some points that are a bit off. And still, you try to remove most of them, but you can't with every single issue where there's something going on. There's still points that there may be something going on, but there's no statistical reason to remove them because they're not. There was no rain and it was not flagged as being bad. So that's kind of I guess a data management question is when is data anomalous enough to justify removing it? Yeah, the eddy covariance system, it'll do stuff like that. It takes a bit of work getting used to it, and it's a bit of a fickle instrument.
Okay, anybody has another question? Okay, if that's all, then thank you very much, Dorothy. This was very clear talk and very interesting.
Yeah, thanks for having me.
Yeah, it's very much in line with what we do in my group.
Okay, that's good.
Yeah, maybe we can discuss that a little bit afterwards.
Yeah.
Thank you everybody for participating and for arriving on time.
Yep.
I'm going to stop the recording now.
Thank you.